I'd like to take just a moment again to welcome our next speaker and introduce Henry Chapman. Henry received his PhD in physics at the University of Melbourne in '92 and did some postdoc work for a couple of years, I believe, at Stony Brook and maybe some work at Brookhaven at the same time, but joined Lawrence Livermore Laboratory in '96. And it's really an interesting time for what became very, very important experiments to be done on X-ray, well, FELs and X-ray FELs, soft and hard. So Henry, shortly after arriving at Livermore, began to collaborate and assemble a team. And that team brought together people who were knowledgeable in things like injection systems and electrospray, in theory, in FEL and damage physics, in biophysics, and really assembled a powerful team of people who started to develop the concepts that evolved in imaging, using coherent imaging, using FELs. And it really laid the groundwork for what I'm going to let Henry, of course, say himself, but this novel idea and the demonstration of this idea of diffraction before destruction evolved from that work. And it was demonstrated at Flash in DESY, and then along came LCLS in 2009. And at that point, Henry again took leadership. He, at that time, had just moved to DESY, and really brought together a large international collaboration. I don't know the number of people, but it was up in the 80 or thereabouts range. Probably took a lot of sociology just to get them to really work together effectively, but he did that. And really evolved this activity into a pioneering research effort, which I think, arguably, but I certainly believe, is one of the most high-impact areas of LCLS research today with tremendous potential, and that is what has come to be known as serial femtosecond nanocrystallography. So Henry's research has been widely recognized. He's Bragg Medal, Edward Teller Fellowship, the Bjorn Vik Prize among them. He did, as I said, join DESY in 2008. He was one of the founding directors of CFEL, the Center for Free-Electron Laser Science. And so I'm very pleased to welcome Henry, who's going to talk on single molecule imaging.
Well, thank you very much, Keith, for that very nice introduction. And it really is a great pleasure to be here today at this 50th anniversary celebrations. I'm really honored to be able to represent this team of people here. And I was really happy to accept this invitation because it gives me an opportunity to really express the gratitude of all of us for providing such a wonderful facility, wonderful atmosphere to perform this really great science. And so I guess to follow on from the theme that Zhirong mentioned, that how new instrumentation, new machines can really generate new ideas. This idea of single molecule imaging at the atomic scale really didn't come about until FELs themselves. The idea of the FEL was proposed and was out there, that you could have femtosecond pulses with such huge numbers of photons that you'd be able to get a measurable diffraction pattern from just something perhaps even as small as a single macromolecule. So it was the FEL that came first, and then the science kind of adapted to that, and it will be of course interesting to see where that goes. But so the idea is to sort of do imaging without lenses, because we don't have any atomic resolution lenses, but it turns out we have really good computers and computer algorithms that can take this place. But we record diffraction patterns such as this one shown here, which has this nice continuous form which can be used to synthesize through a Fourier analysis, synthesize the image, which is sort of the basis of X-ray crystallography in some sense, and also which is the basis for structural biology, which is the way of kind of measuring three-dimensional images of these large molecular machines that drive many of the processes of life. So this is a movie that I got from Paul Nissen in this center at Aarhus University. This is a calcium pump which brings calcium ions across the membrane into a reservoir. The reason this thing exists is in muscle, in the action of muscles. This machine uses about 1/3 of the energy that the human body provides, or is expended in doing this. It pumps calcium ions to enable your muscles to actually relax after they're being contracted. So the calcium ions are taken away from myosin. That allows the muscle to relax. And anyone who's had a cramp knows what it feels like if this thing is somehow short-circuited. But it's this huge conformational changes that occur when an ATP molecule is bound up here, and it induces these conformational changes by breaking down the ATP. And for every ATP consumed, 2 calcium ions can pass through this membrane through here. This structure, I mean, it looks like this beautiful molecular movie here, but it's actually kind of a stop-motion movie because each conformation of this machinery was obtained by sort of chemically isolating that particular state, making a crystal of that particular state, and figuring out the structure, and doing that for several states, and then doing a sort of interpolation using molecular dynamics to figure out what all the motions are. But you can see that you can really unravel how this machinery works and really wonder at the beauty of nature, and coming up with these amazing machines. So the huge conformational changes up here are really just to sort of do things in opening this channel, producing this huge gradient of calcium ions. Now, as I said, this was done by X-ray crystallography, and it's pretty amazing that molecules as big and complex like that can actually be crystallized. You can make them all arrange themselves very nicely into a crystal. At least some of them you can. It can be a pretty tough job, and as I'll mention later on, a lot of effort and tears and frustration is put into trying to make these crystals. But if you can obtain a large, well-diffracting crystal, you can take it to an X-ray machine like a synchrotron like SSRL, record diffraction data such as this, and from this data synthesize these three-dimensional images that I showed you. So what you see here are Bragg spots, and as has been pointed out by Francesco early in the morning, it's the 100-year anniversary of the discovery of X-ray diffraction and its explanation, which occurred in the same year by Bragg's, or the interpretation of these spots. So you can measure the spatial frequency content of the object and synthesize in the computer the 3D image. And so this field is remarkably successful, and that success can be illustrated by this plot here, which shows the number of structures that are deposited into the protein data bank, which is an online repository of all these structures. There's about 80,000 here today, and they're growing at a rate of 7,000 per year. Most of them are actually, well, as shown in blue here, are X-ray structures. So macromolecular structures that have been determined by this method of X-ray crystallography. And this huge explosion of growth, again, is sort of fueled by the technology of X-ray sources, synchrotrons, undulators, dedicated beamlines, robotics to move crystals in and out. And so there's of course, a huge amount of biology and understanding of how all these molecular machines work and how they work together. But the problem is that this need to crystallize, to be able to see the structure, is just a huge bottleneck, and it can take many years of effort, sometimes even over a decade of effort, to try and figure out how a particular molecule will actually crystallize, how to sort of find the conditions to make these nice, very large, well-diffracting crystals. So you may say, well, why do you need large, well-diffracting crystals? Because oftentimes, or very often, you can quickly find very small, less well-diffracting crystals. And the reason for that was explained by Paul Alivisatos this morning. It's due to these scaling laws, and due to the density of defects and things that limit crystal growth. So why can't we just take a small crystal and just expose it for longer and be patient? It turns out you can't be patient because of the problem of X-ray damage. So X-rays, of course, well, we need to use X-rays to get the high resolution, short wavelength that can resolve interatomic spacings. But this means that the X-rays are high enough energy to ionize material. That ionization causes damage to the very structure that we're trying to look at. Here's a series of images on this is a crystal of virus particles, and these are just half-second exposures of a 20 by 20 micron beam, which has moved from place to place, and you can see immediately that there's huge damage incurred in the structure, and after this half-second exposure, the crystal just doesn't diffract anymore in that region. This was done at room temperature, and the actual dose that you can tolerate, dose here is given in units of Gray, which are joules per kilogram of energy absorbed into the system. So you can, this crystal can withstand about 300 kilogray of dose before this destruction happens, and you don't get any more diffraction. You can extend this a bit by going to cryogenic temperatures, since that limits diffusion of free radicals, and so you limit some of the processes of chemical degradation that happen. But still, this, even at 30 megagray, it's not really much exposure. And so you need a really large crystal to give you a very strong enhancement of the diffraction into these Bragg peaks. And the strength of that Bragg diffraction goes linearly with the number of unit cells in the volume of the crystal. And so that is why you need these big crystals to get a measurable signal before you sort of run out of your dose that you can use on that crystal. Now, with the idea of free electron lasers came this idea of actually outrunning radiation damage by using pulses that are shorter than the time scale for these damage processes to occur. So early molecular dynamics simulations that were done by János Hajdú at Uppsala University and Richard Neutzer, also Edgar Vekkerts here in the audience, was one of the authors of this seminal paper, suggested that if you had pulses on the order of 20 femtoseconds or even shorter, you could indeed outrun these processes. You're relying upon the fact that that atoms have inertia, so even with such a huge force inflicted on these atoms due to the Coulomb repulsion that occurs once you remove the electrons through the ionization by X-rays, even with all that going on, it would take some time for these atoms to start moving from their initial positions. And so if you can do this sort of in a flash photograph you would overcome this damage process. And this is somewhat similar to the first flash photographs, which were taken by Harold Edgerton, who developed and popularized the use of, he developed the air gap flash bulb. And he did some of the first investigations just of sort of natural things and various common events, but looking at them at high speed. So here's a picture of an archer shooting an arrow, and you can see the arrow has just left the bow there. But if you actually zoom in here, you'll notice the string of the bow has really pinched into his skin. And anyone who's shot an arrow and has had that happen to them knows that this is a pretty painful exercise, right? But actually the signal, which has started off from the nerve cells here and has to travel chemically up his arm into his brain, and that reaction to be expressed through a contortion of muscles in the face, that hasn't yet occurred. So he hasn't yet registered the fact that this string has really pinched into his arm. So this is an example of outrunning the consequences of the fast event. In fact, so this guy, I mean, he was inexperienced. He was a student of Edgerton. And in following photographs, he's actually wearing a protection on his arm. so he sort of learnt from his lesson. And so just as Harold Edgerton's flash photographs, we want to put as many photons as we can in a short amount of time. We need the photons to get the bright photograph, and we need it in a short amount of time to outrun this radiation damage. And that is exactly what free electron lasers give us. They give us this, as Zhirong pointed out, 9 or 10 orders of magnitude higher peak brightness, which is just, I mean, that's just a huge, insanely huge number. And it really just opens up a whole new ways of exploring nature and looking at structures. So the question is, does this work? And one piece of evidence, I guess, that it does work is that I'm standing here before you, because I probably would here if it didn't work. So I guess it does work. Another piece of evidence is just seen in this diffraction pattern here. We actually have millions, well, hundreds of millions of diffraction patterns that we've now recorded from various different samples. And we could take almost any one of those and sort of look at it and say, yes, look, it works. In this case, this is a diffraction pattern, a single pulse diffraction pattern from a very small nanocrystal of a membrane complex, photosystem 1. And the fact that there is a Bragg peak somewhere out at 3 angstrom resolution means that there was this cooperative enhancement from unit cell to unit cell. Every molecule in this lattice or in this arrangement has diffracted, and this has led to this constructive interference. That would only occur if every structure, every molecule was pretty much the same or exactly the same to this 3 angstrom level. If damage had occurred and all the atoms had been displaced in random ways from unit cell to unit cell, then you wouldn't get this enhancement and you wouldn't see a Bragg spot. So this shows that at least for some part of the pulse, this crystal was indeed a crystal and gave rise to this crystal diffraction pattern. So that just shows us immediately that this works. This was done for this very large protein complex, photosystem 1, which is involved obviously in photosynthesis. This was provided by Petra Frommer, who was very much part of these experiments, and she had worked on photosystems, photosystem 1 and 2, and it had taken her 13 years of effort to get the large, well-diffracting crystals that she had taken to a synchrotron to get the structure, and it's that structure that's shown here. It's a a large antenna complex, essentially, that efficiently absorbs light and transports an electron across the membrane to do the synthesis, in this case, of carbohydrates and things, eventually. So it took her 13 years of effort, but to make very small nanocrystals, she was able to sort of work that out within a matter of weeks. These are much easier to come by. So once we have objects like this, the way we record these patterns is to have them in a fluid, in the fluid that you crystallize them in, and flow that fluid across the beam in this jet as shown here. And then it's just constantly flowing. X-ray pulse comes along, and the diffraction is recorded on these detectors, two detectors as shown here. These are the Cornell SLAC pixel array detectors that can read out frames at 120 hertz, which matches the repetition rate of LCLS. Now, from the point of view of the X-ray beam, looking sort of if the X-ray beam is focused down on here and coming into the page, then just like Harold Edgerton's photographs, the pulse freezes all motion. So it you don't see the flow, obviously. And if there is a crystal in that area at the time that the pulse comes, then it will give rise to this diffraction. Now, the crystals are, as I said, just in this fluid. Here's Petra Frommer. She's pipetting this into the reservoir that we use to inject. There's John Spence ably helping. And he was in his lab. He and his colleagues developed this jet, which you can see here. This is a microscopic image made at the experiment. So this bright spot is a plasma formed by the X-rays hitting the crystals and the fluid, mainly water. And it's glowing because it's all the electrons and ions are flying everywhere and creating a plasma. That was flickering a bit because pulses are coming 120 times per second, but between each pulse, this is moving fast enough that it actually goes several centimeters between these pulses. So you're always replenishing the sample with a new crystal to be destroyed. So we can just measure these things at a large rate. We obtain hundreds of thousands of patterns in a usual experiment where we collect data from a particular sample. And in fact, there's so much data that it sort of gets a bit overwhelming and a bit hard to deal with. There's, of course, no way to look at every pattern. There's just so much of it, and it comes pretty fast, and it comes, so it's quite fast. One way to sort of visualize what you have is just to accumulate it all on the detector, which is okay, and it does reveal some interesting things. Like in this case, there was some actual texture in here, some alignment of the crystals with the flow, because they were needle-like. But when you do this, it loses a lot of the information that you have. It's just projecting it onto 2 dimensions. Actually, what we want to do is really keep the full sort of 3-dimensional glory of all our data. But the trouble is our crystals are just coming along. They're just tumbling along in some random orientation. There's no a priori way to know what that orientation was. But we need to know what that is so that we can figure out how to arrange all this diffraction data, which is describing the 3-dimensional structure of our particular protein. And when we synthesize this image, we have to sort of figure out this 3-dimensional way that each diffraction pattern is cutting through this full description of the diffraction data. But we can do this quite easily in the case of crystalline data because we have this nice lattice to sort of orient ourselves with. So because of the regularity of the crystal itself, the diffraction data, these Bragg peaks are also regular, and they have specific regularity in each direction or a specific spacing. So we can examine the spacing and figure out which Bragg peak is which, essentially assign these Miller indices to every Bragg peak. And of course, this has to be done automatically because there's so much data. There's terabytes of data for each data set. But once you do that, you can do this combination, and you end up with this three-dimensional data set, what I like to call a three-dimensional powder diffraction pattern, because we've essentially analyzed this powder or suspension of crystals one grain at a time. Every individual spot here has a contribution to it from thousands of individual crystals. Each spot is a different subset of 1,000 or so crystals. The ones that happen to be at a particular orientation to diffract into that particular Bragg spot. Now at this point we have essentially the same type of data that a crystallographer is used to. In fact, it's better data because we don't have the problem of radiation damage. But we can now apply the same sort of crystallographic methods to do this Fourier synthesis of the image. In this case, it's done by a process called molecular replacement, where you start up with a structure that's pretty close to the one that you're trying to solve. In this case, well, we had the synchrotron structure to deal with. So you can do this refinement of the structure so it gives rise to a calculated diffraction pattern that matches what you've measured. So this works very well. This is done using Axel Brunger's Den refinement by Axel Brunger. And we see these transmembrane helices. This is looking side-on to the photosystem molecule. You can see all the chlorophylls and carotenoids and things in here. And the electron density is very well defined, and it's for a protein crystallographer, they see this as an extremely good data set. We can even do this at longer wavelengths. This was to see what happens at even higher doses. At the longer wavelengths, X-rays are less penetrating, so this means they actually deposit more energy into the sample. And so the dose in this case was 3 gigagray per pulse. This is 3,000 times higher than what you can actually tolerate at room temperature at a synchrotron. So a huge increase, or 100 times higher than what you could achieve if you were to completely freeze your sample, which could introduce artifacts. Now, another interesting example here is from a enzyme involved, or from a parasite, a nasty parasite that causes African sleeping sickness. It's quite a bad disease in the tropics and fairly neglected because there's not very good medicines for it. This was actually the people who were looking into this disease were expressing this protein by a sort of common way, or overexpressing it in these insect cells by using a virus vector to put out a lot of protein. And the graduate student who was in charge of this, he looked at the cells and actually saw these little needle things, which turned out to be crystals of the protein in particular, and they were formed just spontaneously with no one telling them to crystallize. They just happened. Of course, they're small because the cell is small; it can only produce so much stuff. But the interesting thing is, what was found is that these actually, the protein is modified in the same way that it would be modified in the cell of the parasite when it produces these things, which is to inhibit the action of that particular protein. And it does other translational modifications, like adding these carbohydrates and things, which are not part of the amino acid sequence. They get sort of bonded on after the fact. So this actually gives you the answer, or a way of figuring out how to actually inhibit this particular protein. Perhaps gives you ideas to how to design a drug that will stop the action of this protein, which has to be done in a careful way because you don't want to inhibit the human's version of this particular protein. So that's the combination of this in vivo crystallization with FELs that can deal with the very small crystals that are produced is an extremely powerful one and could be become quite important. Now we know that the crystals are small because we can actually measure directly from we can count the number of unit cells. These are the Bragg peaks. Now we're zooming in quite strongly into this, and we can see these fringes here. This is due to the truncation of this lattice. It's not an infinite lattice, which you usually assume that you have in crystallography, but you can actually sort of count the number of fringes, which actually tells you the number of unit cells. In this case, about 15 of them. Why is this good? Many reasons. One is that you can actually see here intensity that's not just limited to the Bragg peaks. And this is important, as shown here in this very quick tutorial about X-ray diffraction. In a crystal, you have this repeating molecule over and over again on some lattice, so you're convolving this molecule with the points of lattice. That means when you form your diffraction pattern, you've got the diffraction pattern of the single molecule, so it's like a single molecule diffraction, but now modulated or multiplied by the reciprocal lattice, which again is just points. And this is usually quite restrictive. There's not enough information here to actually fully carry out this Fourier synthesis. But if you could measure halfway in between the Bragg peaks, it would be the same as pushing your unit cell out to be twice as big, but leaving the same molecule there, which means you now have all this empty space here, which you can use, because you know it has to be empty, you can use to constrain the phases so that you know how to do this reconstruction. This was first put forth by David Sayer, actually 10 years before SLAC was formed, so in 1952. This is the entire paper reproduced here, so this is not the abstract, it's the entire paper, half a page. And it was, it was very insightful. You can read it, but actually I don't have time, so it's better to show a picture of David Sayer. This is him on his 80th birthday, which was held here in 2004 in a workshop that we held. So Keith Hodgson, Janusz Hajdu, and I ran a workshop here. This is at the Redwood Room. And it happened to be David's 80th birthday, and Keith gave him this nice cake, which we all ate, and it was a very nice occasion. Sadly, David passed away this year, and so we lost a great colleague, which was very sad. But actually, this here is John Miao, who was a student at Stony Brook, working with him, who first, with David and Janusz Kurtz, demonstrated this idea of actually using the continuous coherent diffraction pattern, which has no Bragg peaks because this thing is not periodic, and using these ideas from crystallography to use a sort of computational lens to do the imaging by the Fourier synthesis of this diffraction pattern. Now, getting back to our Bragg peaks now, we'd like to measure in between them, and actually from these very small crystals, we can indeed see things all over the place between all the Bragg peaks. So now suddenly we've got this new handle on examining the molecular transform of these particular crystals. There's information just all over the place. We've still got to figure out exactly how to use it, because we've got to get rid of the influence of the Bragg peak itself, but we have some ideas, actually, that John Spence originated. So what we'd like to do is use that to get this underlying molecular transform. We actually haven't done it. This is a cheat because we know the structure, which was solved by the traditional molecular replacement method. But it's very enticing, and it's actually very close to being able to do this, I think. And so then we'll be able to solve structures of native proteins without having to have either a known structure or some other method like some chemical replacements of putting in heavy atoms and things like this. Now, we have also, of course, looked at single particle diffraction, so again, without any Bragg repeatability, and we can reconstruct these images very robustly and very easily because we have such high coherence with the X-ray beam, and the data is just so good. I mean, it's just, I mean, most people looking at these things, they say, is that a calculation or is that the measurement? And these are real measurements. And in fact, we've applied this to an interesting problem of imaging soot. This is a project that was led by Mike Bogan, who's here at the Pulse Center. And using now, instead of a liquid jet, a kind of smoky beam of soot that's brought across the X-ray pulses in the same way. Now, soot is hard to examine otherwise because at high resolution. To look at it, otherwise you'd have to sort of put it onto a surface and put that into an electron microscope, which could totally change the structure of this very nebulous thing that you're trying to look at. This is obviously important in climate research, also in health, to understand how these things get into your lungs, for example. So we've measured many diffraction patterns from many different objects. And they all are different, and they have quite varied, quite unexpected morphologies. We can also examine just in reciprocal space and look at things like the fractal dimension. Now, soot is all rather unique structures. Each one is different to the other. Because we destroy it on each pulse, we can only see one view of it. Now, when we have reproducible structures, like virus particles, this is a Mimi virus, which is a very large virus particle, then we can collect diffraction data of all these various orientations again from the same structure and do the same sort of three-dimensional orientation of the patterns as we do with the crystals. The trouble is now we don't have this nice Bragg peaks that sort of act as signposts to tell us how things are oriented. So we have to correlate data with itself, and it's a much harder problem, but it has now been solved by various nice algorithms. So once you do that with the data, then you can reconstruct a three-dimensional density map of this particular virus. This is still at fairly modest resolution of 50 nanometers, so it's not showing much detail, but it is showing more detail than you could achieve even with an electron microscope for these large things because the electrons don't penetrate. This reconstruction was done by Thomas Eckerberg at Uppsala, and this particular project led by Janos Haidu. But we'd like to really push this down to much higher resolution, much better resolution, and one way is to have these nanocrystals and go to smaller and smaller and smaller down until the single unit cell, and smaller virus particles. Of course, the challenge is that as we go to these smaller things, the signals are getting much weaker, and we have to learn how to deal with diffraction patterns that have much less than a single photon per pixel per shot. This is a simulation of what you get from this virus particle, which is a sort of smallish virus particle, but still it's a very weak diffraction pattern. So what we need in this case is to put more photons onto the sample. We know from our experiments and modeling that when you do that, the explosion does happen a little bit faster, so you need to put those pulses into an even shorter, put those photons into an even shorter pulse. So we need these developments that Zhirong was talking about, like getting to the terawatt pulses, because it's intensity, it's photons per unit time, which is important here. But we're trying a whole lot of other tricks, of course, to get to the single molecule or just molecular structure. So the nanocrystals, of course, are very successful. We could think of other ways, for example, orienting single molecules say with a laser field, which may be something that the next speaker would be well equipped to do, or other tricks such as shown here. Actually, I'll just quickly show this before I run out of time. This was another way of accumulating data from very weak patterns. So in this case, it is to form, from every diffraction pattern, form a sort of self-correlation function, an angular correlation function from each diffraction pattern. This angular correlation function is actually independent of the orientation of the particle. So no matter how the particle is oriented, you can do this correlation, and it will just pick up the internal symmetries of that object itself, or the symmetries of that object. You can then accumulate that and give rise to this four-dimensional correlation function. Actually, for this particular case, three-dimensional, because of the cylindrical symmetry of these objects that were these little dimers. So this you can just keep adding to pattern by pattern, and when you've got enough, you then need to do an inverse problem to extract now what the actual diffraction pattern would look like in 3 dimensions, and then do a second inverse problem, which is to reconstruct the structure. And this has been demonstrated by Dmitry Starodub here at SLAC in Mike Bogan's group. So I hope to convince you that X-ray free electron lasers really open up a new door and should really, I think, revolutionize structural biology. So even if we're still stuck with these nanocrystals, this is still a huge thing and really opens up many more structures that can be solved by this method. Membrane-bound proteins in particular, things like G-protein coupled receptors, and using the scaling laws that Paul mentioned this morning. So this is quite a large development on itself. We also avoid radiation damage, so the data are very good, and perhaps even better than what you can achieve with a synchrotron. You could imagine doing this at quite high throughput. Everything is sort of fluid-based. You could use microfluidics to shoot samples around, do mixing and things like that. So there's no need to mount specific crystals in the beam using robots or graduate students. Data are collected at room temperature. We've got new ways of doing the phasing that opens new ways of looking at the data. Of course, because we use short pulses, you can also do time-resolved structures. So now we could look at these calcium pumps, for example, or you could look at a drug interacting with a molecule, or many other processes. So this really was a large collaboration, as Keith said in the start. This is just actually a few of the faces. Petra that we saw before, this is Laz Radeke, who was working on the African sleeping sickness project. Richard Neutzer here from the Neutzer paper. Ilma Schlichting was very helpful in making sure we got all the crystallography right. His Bruce Doak, who developed the jets with John Spence, and also Dan DuPont there was doing that. But here's a more complete list of names. Well, maybe it's not complete, and if it's not, I apologize. But so we can't go through them all, but I would especially like to thank the LCLS group for making such a wonderful facility with such great technical support, and really all of SLAC, which helped make that happen, and have made this such a terrific place to do science, which just cannot be done anywhere else in the world. So thanks.
Some questions? Okay, questions, please. Wave your hand. Here, Don Plummet, and then.
Henry, you just have been provoking a question. So you were saying about this radiation damage, you avoid that, and you explained that by, imaginary, you get this X-ray beam on the sample, it rips off the electrons, but the ions are so sluggish that it takes time until they really cool or explode. But since the X-rays are scattering from the electrons, now what is actually the real definition of a radiation damage here?
Yeah, so in structural biology, you just need to find the location of the atoms, pretty much. What are the nuclear positions? So not so worried about understanding, say the higher molecular orbitals and things like that, which will not be present in this data. Yeah, so we're relying upon the fact that some of the electrons do stay around, and there are bumps in the electron density which point to the positions of the atoms. And you may even get the electron density wrong, so it may look like a carbon atom instead of an oxygen. But since you know the amino acid sequence and you know sort of what's what and how they bond, this even can be corrected quite easily.
So in fact, in the data refinement, you already see the missing electrons.
Well, actually, we've been looking for this, and it's not so obvious.
Okay.
Back in the open.
What?
Henry, when you want to use the diffuse scattering between the Bragg peaks, I think the key point is also to reduce the background. Now, what are the main sources of background, and are there possibilities, or do you see possibilities to reduce this?
Yeah, one source of background is fluorescence, which you could reduce by filtering the data. If you have single photon counts, then you can sort of distinguish elastic scattering from fluorescent data. But most of it is elastic scattering from, say, the water in the liquid jet. And so one way to reduce that is to have less water, to have thinner jets, and that's been the real key technology driving this, which is to make these very narrow jets by speeding the liquid up to sort of stretch it out using gas. But you could go further and not have a liquid jet sort of aerosolize the whole molecule, but do it in such a way that there's still enough water that it keeps the structural integrity. So it's a bit of a fine balance. Of course, there's all there still is some atomic motion that's going on, especially near the end of the pulse, and that is giving rise to sort of non-structured background. So for that, you should use shorter pulses. And again, so we want to squeeze as many photons into as shorter pulses as possible. And also, if getting back to Helmut's question, if we can go to even shorter pulses that outrun even the, say, Auger decay rates and things like this, would be in a much better situation too, if we need it.
One more question. I don't see where. Okay. Got a mic.
Tony.
Sorry.
Henry, so far, I think for the crystallography, you've had to use molecular replacement. Is that correct?
Yes.
Is there the possibility to go beyond that and relax that sort of constraint?
Yeah, so there's two great possibilities. One was what I was showing, to use in between data and do the phasing that way using iterative schemes. The other is to use anomalous diffraction. And you may think, well, that's never going to work because you're ionizing the hell out of the molecules that you hope to use. But it turns out it could actually get better with the high intensities, that you can actually enhance this anomalous signal. And one way to do that would be to sort of sit just above an absorption edge. Of course, that particular of a heavy atom in the sample that atom's going to absorb a lot of energy because you're above its absorption edge, obviously. So it's going to ionize like crazy. And it's going to ionize so much that it essentially disappears and bleaches out. Its scattering factor sort of bleaches out. And whereas if you go just below the edge, it's going to be a bit more normal. And so there will be a difference for that one particular atom compared to all the other atoms, which are damaging as much as they usually do. And so this will give you a way to actually see these, specifically see where those atoms are and do a kind of anomalous diffraction-like measurement. So we're going to try that out in beam time here in October, actually. So, yeah.
Okay, let's thank Henry again.